Good afternoon one and all. So, here we are with our last segment of cell structure, wherein today we are going to discuss in detail about double membrane bound organelles, mitochondria, chloroplast, nucleus, chromosome structure, shapes of the chromosomes, and lastly microbodies. Let us get started with the first double membrane bound organelle, that is mitochondria. Mitochondria, if you have to describe its structure, we can say mitochondria as a cylindrical sausage shaped organelle. Okay, mitochondria is a cylindrical sausage shaped organelle, wherein mitochondria diameter of the organelle will be around 0.5 micrometers in diameter and length is around 1.0 to 4.1 micrometers in diameter. This will be the diameter and length of mitochondria. Fine. And how can you describe the structure of mitochondria? Mitochondria is double membrane bound, wherein outer membrane of the mitochondria is continuous and limiting. Okay, outer membrane of mitochondria, that is outer mitochondrial membrane is continuous and limiting boundary of this organelle. Okay, this is continuous. And if you have to look into the protein and lipid composition in the outer mitochondrial membrane, we can observe 40% of lipids and 60% of proteins. And outer mitochondrial membrane is completely permeable. Okay, this is completely permeable. Fine. And then is inner mitochondrial membrane. Fine, ma. Inner mitochondrial membrane is also called as cristae because of its enfoldings. Okay, inner mitochondrial membrane is said to be cristae because of its enfoldings. So, here if you see the protein and lipid composition, proteins will be around 80% of proteins and lipids are 20% of lipids. 80% of proteins and 20% of lipid composition is present in the inner mitochondrial membrane, that is cristae, and this is semi-permeable. Cristae is semi-permeable or selectively permeable. Semi-permeable or selectively permeable. Take down parallelly, ma. Yeah. And the space in between the outer mitochondrial membrane and inner mitochondrial membrane is called as intermembrane space. Okay. The space in between these two membranes is called as intermembrane space. And in inner mitochondrial membrane, that is cristae, all the electron carriers, protein channels which are required for electron transport system are present. Okay. All the electron carriers, protein channels required for ETS are present in the or are embedded inside the inner mitochondrial membrane. And F0 F1 particles, F0 particles which are also called as ATP synthase or elementary particles, these are also embedded in the inner mitochondrial membrane, that is cristae. Okay. So, outer mitochondrial membrane, inner mitochondrial membrane, its protein and lipid composition, what are embedded inside the inner mitochondrial membrane, and what is intermembrane space. This is clear to you, right? Now, mitochondrial matrix. What is present inside the mitochondrial matrix? So, here if we have to see mitochondrial matrix, in mitochondrial matrix you can see double stranded circular DNA, double stranded circular DNA, few RNA molecules, few RNA molecules, few RNA molecules and some proteins. Okay, double stranded circular DNA, few RNA molecules and proteins, and the components like ADP, inorganic phosphate and oxygen are present inside the mitochondrial matrix. So, these are the components and composition which you see in the mitochondria. Okay, mitochondria, which is double membrane bound organelles, outer mitochondrial membrane, inner mitochondrial membrane, what is present in the intermembrane space, and what are the components that are present in the mitochondrial matrix. This and all are important. Okay, now we will be looking into what are the functions of mitochondria. Okay, mitochondria is keenly involved. The primary function is beta oxidation of fatty acids or beta oxidation of the biomacromolecules. You can say beta oxidation of fatty acids, and it is keenly involved in aerobic respiration, fine, and it is involved in the synthesis of ATP mainly, and this stores ATP. That is why mitochondria is called as energy currency of the cell. Okay, as it is storing the cellular energy, that is ATP. ATP is called as cellular energy because it is the simplest form of energy where the cell can directly utilize ATP to conduct all its metabolic activities. So, ATP is what? Cellular energy, fine, which is stored, which is synthesized and stored inside the mitochondria. That is why mitochondria is called as ATP is the energy currency of the cell, and mitochondria is called as what? Powerhouse of the cell, which is the energy currency of the cell, and mitochondria is called as powerhouse of the cell. Fine. Now, we will be discussing about F0 F1 channel. So, in the inner mitochondrial membrane, F1 is present, which is embedded inside the inner mitochondrial membrane, which will pump the protons from intermembrane space to mitochondria, to mitochondrial matrix. Okay, where F1 is embedded inside the inner mitochondrial membrane and F0 is placed towards the matrix. Okay, remember that F0 F1 pump which is present, where F1 is embedded inside the inner mitochondrial membrane and F0 is towards the matrix. Okay, this will pump the protons from intermembrane space to the mitochondrial matrix. Clear, right? This is all about the organelle mitochondria, which is a double membrane bound organelle. Second double membrane bound organelle that we have to learn is plastids, of which we will highlight the chloroplast. Okay, plastids. We have three types of plastids that are observed in plant cell. Okay, plastids are purely present in the plant cell. We have three types of plastids. First type is what? Chloroplast. Second is chromoplast, and third type is leucoplast. Fine, chloroplast, chromoplast and leucoplast. Chloroplast is the double membrane bound organelle which has fat soluble pigments. Okay, like it has fat soluble pigments like xanthophylls, carotenoids, chlorophyll. Chlorophyll is abundantly present inside the chloroplast, and chloroplast number may vary depending on the plant cell. If it is a primitive plant cell like algae, Chlamydomonas, where a single chloroplast is present per cell, okay, where chloroplast is cup shaped inside the Chlamydomonas. But then in a mesophyll cell of higher plants, you can see 20 to 40 chloroplast organelles present per mesophyll cell. So here if you see, chloroplast organelle can be 1 per cell in Chlamydomonas. You can observe 20 to 40 per cell, 20 to 40 per cell in primitive algae, in advanced or higher plants like what? Mesophyll cells. Clear, right? This is all about chloroplast. Now chromoplast. Chromo means color, plast means plastid. Colored plastids are chromoplast. Different fat soluble pigments like xanthophylls, carotenoids, anthocyanins will produce different pigmentation of leaves. Even in leaves you can see diverse variety of colors. That is because of what? Chromoplast. And last is leucoplast. Leucoplast are those plastids which just store the food material. Okay, they are colorless and they store food materials. Based on the storage product, we have different types of leucoplast. If it is storing starch, then it is amyloplast. If proteins are stored, it is aleuroplast. Fine. If lipids or fats are stored, it is called as eleoplast. Fine. So these are different types of plastids that we have to know for our exam. Okay, important. And then now we will clearly look into the chloroplast structure. It is double membrane bound organelle wherein you have outer membrane and inner membrane. Inside the matrix of the chloroplast, you have membranous sac-like structures called as thylakoids. Okay, membranous sac-like structures. These membranous sac-like structures are called as thylakoids. Membranous sac-like structures are thylakoids. And thylakoid membrane is called as thylakoid membrane or lamellae. Thylakoid lamellae or thylakoid membrane. And the stalk of sac-like structures is called as grana. One grana is connected to the other grana by stroma lamellae. Okay, one grana is connected to the other grana by what ma? Stroma lamellae. Fine. And inside the stroma, if you see, what are the components that you observe inside the stroma? Stroma is said to be chloroplast matrix. Chloroplast matrix or the fluid filled space, that is stroma, is one and the same. This consists of starch granules, lipid droplets, and most importantly double-stranded circular DNA. Double-stranded circular DNA is present both in the matrix of chloroplast and mitochondria. In mitochondrial matrix also you will observe double-stranded circular DNA, where that takes care of the metabolic activities of that particular organelle. But here double-stranded circular DNA, which is present in the chloroplast, is involved in the synthesis of proteins. That is, world's abundant protein is synthesized, which is rubisco. Fine. Rubisco synthesis happens inside the chloroplast stroma, or you can say the matrix of the chloroplast, right. So these are all the things that you have to know with the organelle, that is chloroplast. And then here you can see clearly, right, the chloroplast DNA outer membrane, inner membrane, intermembrane space, lipid droplets and the fluid filled space, that is stroma, and the ribosomes. Most importantly, what type of ribosomes that you observe both in the chloroplast and mitochondria. Both in chloroplast and mitochondria you observe 70S type of ribosomes, where the larger subunit will be 50S and the smaller subunit will be 30S. And you have the membranous sac-like structures, thylakoids, lamellae, membranous lamellae, stalk of sac-like structures is grana, and you have a starch granule. So, this type of ribosomes, 70S ribosomes, are observed in chloroplast and mitochondria. 70S ribosomes are observed in chloroplast and mitochondria, and they have double stranded circular DNA. These two are the characteristic features of what? Prokaryotic cells, which are resembled by two double membrane bound organelles, mitochondria and chloroplast. That is because, and this is clearly explained under the concept of endosymbiotic theory. Endosymbiotic theory, which clearly states that chloroplast and mitochondria are once upon a time, they are the independent prokaryotic cells in the ancestral time. In the process of evolution, these these prokaryotic cells, that is mitochondria and chloroplast, are being engulfed by the eukaryotic cell, and they became part and parcel of the eukaryotic cell. But still they resemble the characters of the prokaryotic cell, that is having 70S ribosomes and double stranded circular DNA. As they are possessing the double stranded circular DNA, both chloroplast and mitochondria are called as semi-autonomous cell organelles. Semi-autonomous or self-replicating cell organelles. Semi-autonomous or self-replicating cell organelles. Okay? Note that point, which is very important. And then last double membrane bound organelle that we have to look into is nucleus. Nucleus is discovered by Robert Brown in the year, nucleus is discovered by Robert Brown in the year 1831. Okay, nucleus discovered by Robert Brown in the year 1831. Where nucleus is a double membrane bound organelle, again it has a nuclear envelope. We have learnt all the four components of nucleus in the yesterday's class also. What are the four components, ma? Nuclear envelope, nuclear pores, nucleolus and chromatin, nucleoplasm. These are the things which are present inside the nucleus. So, the components of the nucleus and the nucleus structure is clearly analyzed after staining through a basic dye, that is acetocaramine. This is a basic dye. After this staining procedure, the structure of nucleus is more elucidated. So, now here if you see, inside the nuclear envelope will continue. First component is what, ma? Nuclear envelope, right? Nuclear envelope. Nuclear envelope is extended to the network of reticulum structures. This network of reticulum structures is called as endoplasmic reticulum. This is clear to you. And over the nuclear envelope, small pores are present, that are nuclear pores. This nuclear pores will enable the exchange of the molecules, particles or ions into the nucleus or out of the nucleus. And third component is what? Nucleoplasm. Nucleoplasm or matrix of the nucleus. So, in the nucleoplasm you can see nucleolus and chromatin. Okay, nucleolus and chromatin are observed inside the nucleoplasm. Now you have to clearly know the synthesis, what synthesis happens inside the nucleus. So, inside nucleus if you see, so inside nucleus if you see, and here is a nucleolus. Inside a nucleolus, rRNA synthesis will happen. rRNA synthesis. that is ribosomal RNA is synthesized by the polymerase RNA polymerase 1, by RNA polymerase 1. And inside the nucleoplasm, if you see, both mRNA is synthesized by RNA polymerase 2 and tRNA will be synthesized by RNA polymerase 3. In nucleoplasm, this is inside nucleus, and here inside the nucleoplasm, what synthesis happens, ma? RNA polymerase 2 will synthesize mRNA and RNA polymerase 3 tRNA. Clear, right? So, these are the synthesis functions and these are the components of nucleus. Now, we will look into the chromosomal packing later. First, we will analyze the chromosome structure clearly. What is chromosome structure? Chromosome structure is clearly visible during which phase of cell division? Okay, most of the times the questions are asked as such. Chromosome structure is clearly visible during metaphase of cell division. It is clearly visible during the metaphase of cell division, where chromosome at the center, fine, at the center of the chromosome there is primary constriction. This primary constriction is called as centromere. Primary constriction or centromere. Most of the chromosomes will also have some secondary constriction at the tip of the chromatids. This secondary constriction is said to be the satellite DNA, which has most of the repetitive DNA sequences. So, this is what? Secondary constriction or satellite DNA. Fine. So, here if you see inside the chromosome, it has two arms of the two chromatids are present over here. In this chromosome, how many chromatids are present, beta? Two arms of the chromatids are present, and each two chromatids are present and each chromatid is having two arms. These are two sets of the chromatids, two chromatids, and each chromatid has how many arms? First arm and second arm. Two arms of the chromatids are present. So, here on total how many chromatids are there? Two chromatids, and each chromatid is having two arms, okay, and there are small disc shaped structures which are at the sides of the primary constriction. The disc shaped structure which is at the side of the primary constriction helps in the attachment of the spindle apparatus, and this helps during the cleavage of the centromere. During centromere splitting, that is in the anaphase, the kinetochores will be attached to the spindle fibers, and that is how the cell division is mediated. So, at each primary constriction, how many kinetochores are present? Two disc shaped kinetochores are present. Kinetochores are present, to which spindle fibers will be attached. Spindle fibers will be attached. Spindle fibers are attached to what part of the chromosome? If that is the question, your answer should be what? Kinetochore. Spindle fibers attaches to the kinetochore. So, this is the structural description of the chromosome. So, now we will also look into the nucleus. Not in all organisms nucleus is present. Not in all organisms or not in all cells the nucleus is, single nucleus is present per cell. Not in all the cells many nucleus are present. So, we have those cells where nucleus is absent, we will call those as anucleate condition, and few in most of the living organisms, be it in human cells, single nucleus per cell is present, and in few cells multinucleated condition is also observed, multinucleated condition. So, anucleated condition where nucleus is absent, this condition you will see in mammalian RBC, mammalian mature RBC cell, not in the progenitor cell, mammalian RBC, and in the sieve tube cells, okay, and multinucleated condition is observed in Opalina, and this is also seen in Paramecium. Paramecium you can observe binucleated condition, 2 nucleus are observed inside the paramecium. So, this is about the nucleus structure, components and functions. And one more thing to highlight about the semi-autonomous cell organelles, that is mitochondria and chloroplast, is they divide by amitosis. They divide by amitosis or fission. That is, during the organelle division there is no involvement of spindle apparatus, they will just divide as such. So, the division is through what? Fission or amitosis, and they are possessing 70S ribosomes, and they also have double stranded circular DNA. These are the characteristic features of prokaryotes which are also resembled by these two organelles. Clear, right? Now, this is about nucleus and about chromosome structure. Now, we will look into the shapes of the chromosomes. Shapes of the chromosomes, if you see, we have 4 different shapes of the chromosome, and shapes of the chromosomes are clearly visible during anaphase of cell division. Okay, during which phase? Structure of the chromosome is clearly visible in the metaphase of cell division. Shapes of the chromosome is clearly visible during the anaphase of cell division. So, here if you see, first 2 equal arms of the chromatids, that is the metacentric chromosome. Okay, metacentric chromosome. 2 equal arms of the chromatids are present, and how this will segregate towards the opposite poles in V shape. This shows V shape as the arms are equal. Next, and here the centromere position is exactly at the center in between 2 arms of the chromatid, and here if you see, one short arm and one long arm, and here centromere position is close to one arm and far from other arm. So, here it is showing what? This shape of the chromosome is called as what? Acrocentric, submetacentric chromosome. Submetacentric. One short arm and one long arm. Okay, and this gives out to be what shape? L shape. Okay, one long and one short arm. And next shape of the chromosome is acrocentric chromosome. Acrocentric, where you can see one very short arm and one very long arm. And what will be the shape of the chromosome here? J shape, one very short arm and one very long arm. And lastly, it is telocentric chromosome. Okay, telocentric chromosome. Telocentric chromosome, you can see one negligible short arm and one very long arm. Okay, one negligible short arm and one very long arm. This is telocentric chromosome, and the shape is I shaped. Okay, like this. One arm is negligible. So, these are about shapes of the chromosomes, where you will clearly observe this during the anaphase of cell division. And lastly, about microbodies. What are microbodies? Small single membrane bound organelles which are present inside the cell. These are the microbodies. We have 2 different types of microbodies, peroxisomes and glyoxysomes. Microbodies, how can you describe them? Single membrane bound organelles. These are present both in the plant cells and animal cells. They absorb what? Molecular oxygen, and they will take part in the oxidation. Okay, they take part in the oxidation of macromolecules, especially beta oxidation of fatty acids, by absorbing the molecular oxygen. Next, microbodies are of 2 types. First type is what? Peroxisomes. These are covered by single lipid bilayer membrane. Okay, they are covered by single lipid bilayer membrane, present both in plant cells and animal cells. What are its key functions? Fatty acid oxidation, lipid synthesis, detoxification, glyoxylate cycle, and most importantly photorespiration in C3 plants. Photorespiration in C3 plants. Next is glyoxysomes. Glyoxysomes play crucial role during what? Seed germination. Okay, these are the microbodies which are specially present in the plant cells. They play key role in the seed germination time. Okay, in their keenly present in the plant cells. They play special role during seed germination. So, these are the organelles where we have completed all the set of organelles. Lastly, it is microbodies, that is peroxisomes and glyoxysomes. So, what all we have learnt from cell chapter? We have learnt completely from the chapter cell tissues. We are going to start from tomorrow. From complete cell structure is done till today. We have learnt initially about the prokaryotic cell and eukaryotic cell, both plant cell and animal cell, where we have clearly elucidated the A membrane bound organelles like ribosomes and centrioles of animal cell, and then we have clearly looked into the endomembrane system, where we have clearly analyzed the structure and functions of 4 organelles. Those are endoplasmic reticulum, Golgi bodies, lysosomes and vacuoles. Post that, we have looked into the double membrane bound organelles, mitochondria, plastids and nucleus, and then we have completed the shapes of the chromosomes, structure of the chromosomes, and lastly what? Microbodies. So, with this the cell structure is done. In our next class, we will be looking into the cell cycle and cell division, both the interphase and M phase, mitosis and meiosis. We will deal it in a detailed way. So, stay tuned, and that is all for today's class. Thank you.